Ladies and gentlemen, on behalf of Invivoscribe and the EHA conference, we want to express our appreciation to each of you for participating in this virtual workshop session. We also want to thank our three speakers for their well-prepared and very interesting presentations. We have some time now where we're able to present some questions live to our speakers, which have been sent in from the participating audience. The first question today is targeted to Dr. Arcila. Let me go ahead. Dr. Arcila, in the presentation was mentioned that you are using 700 nanograms of DNA in duplicate for testing and monitoring of samples. Is that enough DNA to reach the mentioned sensitivity of 10 to the minus fifth?
Yes, actually we have been using 700 nanograms in duplicate for 2 years now. In each of the runs, we do have negative controls and MRD controls to track different clones at different levels. So we specifically have controls that are at 1 times 10 to the minus 4, 1 times 10 to the minus 5, and 1 times 10 to the minus 6. And we can consistently show in every single run that we have been able to detect that control over 99% of the time, the 1 times 10 to the minus 5. Now, when you get to 1 times 10 to the minus 6, it definitely goes down, and we're only able to detect it anywhere from 60 to 70%. But we are confident, based on the controls that we always use, that we are hitting that level of sensitivity.
Very good. Thank you very much. The next question I have is for Professor Galimberti. The question is, you presented that the targeted sensitivity for NHL, ALL, and CLL is 10 to the minus 4, and that for multiple myeloma, it is 10 to the minus 5th. Why is it different?
Because the study, as I showed in one of my slides, different studies in multimyeloma approached MRD with 10 to minus 5 because they used a particular NGS kit that simply reached this. This is a tradition, if you want, but as I showed, 10 to minus 6 seems to be more predictive than 10 to minus 5 and then 10 to minus 4. Maria clearly showed that myeloma is different from other hematological disease because she showed that flow cytometry and molecular biology are quite superimposable. So 10 to minus 6 is really different because it is not reachable for flow cytometry, and the ongoing studies will use this cutoff. It's more predictive.
Thank you very much. The next question we have is for Professor Tiemann. Professor Tiemann, this looks like a two-part question. The question is it possible to determine if a single clone is present with two rearrangements or if two unique clones are detected? And also, what are the considerations for CLL in which one clone is mutated, but a secondary clone is unmutated?
That's indeed a very good question. The last part I cannot answer. That is experience. My impression was if there is an unmutated clone, this is the more dangerous clone. So the unmutated clone counts. And the first part, you can solve this problem by allele frequency. If you see the same allele frequency, you can say this is a biallelic rearrangement. And if the clones are of different size, you can consider this is a combination of clones, two separated clones.
Okay, very good. Thank you. Dr. Arcila, For B-cell lymphomas, you were able to detect B-cell lymphoma cases using NGS only. Why weren't any cases detected by flow cytometry only, as for the other diseases tested?
So this really depends on the protocol. We have the protocol that we use for plasma cell neoplasms, for example. It reaches a much higher sensitivity than for the B-cell lymphomas. For the B-cell lymphomas is anywhere in between 1 × 10⁻⁴ and 1 × 10⁻⁵, and it also, it depends on the number of colors that you're using and also the number of events that you're acquiring. So in our experience with the current protocols that we have for flow cytometry, we really didn't have any cases that could be detected by flow cytometry only based on the protocol that we have. However, for BALL, for example, we did have, if we go back to that slide, we did have a few cases where only flow cytometry detected some of the clones, and this actually had to do with the development of new clones. Which is not something that we see in the B-cell lymphomas, but you can actually see in the BALL. So that was 2 factors that were responsible for the findings that we had.
All right, very good. Thank you so much. The next question we have is for, again, Professor Galimberti. Professor Galimberti, you showed data generated from cfDNA with LymphoTrack. Is this possible by using LymphoTrack without any modifications, and which targets were tested?
Yes. We tried the LymphoTrack kit either on exosomes or on cell-free DNA. We used a particular kit for exosomes. There are 2 different kits, one for extracting RNA and one for DNA. We use that for DNA, and that we use the LymphoTrack kit as for normal genomic DNA. And we also extracted in DLBCL cell-free DNA. And in an interesting case, we saw that NGS predicted the relapse before bone marrow, peripheral blood, and only cell-free DNA was predictive as reported in literature. So it's function.
Very good, thank you. The next question we have is for Professor Tiemann. Professor Tiemann, it seems that CLL examples tend to have higher clone frequencies with a lack of or reduced polyclonal background when compared with some of the other diseases. Is there a specific cause for this, or are there insights to be gained from a lack of polyclonal background?
Also a very good question, very interesting detail. But my impression is that in other lymphoma, there are many companion T cells and other cells, and it's a little bit depending whether you look at peripheral blood or if you look at lymph nodes. And this should always be compared with conventional cytology, so with a peripheral blood smear, or with the lymph node histology, so that you can decide how large is the amount of neoplastic cells probably, and how high is the number of accompanying reactive cells. And then you can plausibilize this mixture of cells. Always it's not a cell culture. You always look for maybe natural mixture of reactive lymphatic cells and monocytes with neoplastic cells.
All right, thank you. Professor Tiemann, it looks like you get the next question also, so you have 2 in a row here. Thank you. So Professor Tiemann, how did you determine the stereotype subsets?
Oh, the stereotype subsets, I think this is shown by the rearrangement lists. You have to look up the V and J regions, and then you can identify which type of subset is included, and then you can. It's very easy to read in the table.
Okay, excellent. Thank you for that. All right, I'm just looking to see if we have another question coming through. Yes, we do. Dr. Arcila, what are the reasons behind the fact that some residual diseases in monitoring samples have been detected by flow cytometry only and not by NGS?
So this goes back, is very similar to the prior question. So in some of the cases for plasma cell neoplasms, for example, the reason why they're detected by flow cytometry and not NGS has to do with the fact that the assay is just incredibly sensitive. We capture about 5 million cells. And that sensitivity is not possible with the amount of input that we're using right now for the NGS assay. However, we do have some cases, and again, this is particular to the BALLs, where you have clones that are detected by flow cytometry as being disease, but those clones actually do not have the same clonal sequence, and they have a different clonal sequence. And we have actually monitored those patients, and while we're not able to call them at low level when they are first appearing, we are able to call them later on as a new clone that was not detected simply because we were not looking for this specific sequence by NGS, but flow cytometry is detecting it by the surface markers. This is actually a very rare event. Most of the malignancies that we have seen do not have a new clone, and the clone is trackable. But there are some rare instances where the clone is different, and is usually patient becomes resistant to treatment, and one additional clone evolves. And while it becomes high enough for you to detect it as a new clone and to call it a new clone, it may take a little bit.
Can I add a comment about the question? Thank you. You must put attention also to the bone marrow needle, because if you sample bone marrow before for flow cytometry, and then you take off bone marrow sample for molecular biology, perhaps the sample is not the same. The second one sometimes is more diluted. So also in some labs, biologists mix the sample for flow and the sample for molecular biology in order to avoid dilution for one technique instead of the other one.
Very good. Excellent. Thank you so much for the addition. Because our time is now completed, we'll bring this workshop to a close. We thank again to everyone for their attendance. We would also like to once again thank our three speakers for their fine presentations and their insights in answering a variety of questions. A big thank you goes out to our moderator and speaker, Dr. Maria Arcila of Memorial Sloan Kettering Cancer Center in New York City, Professor Sara Galimberti of the University of Pisa, Italy, and Professor Markus Tiemann from the Institute for Hematopathology of Hamburg, Germany. This workshop has been sponsored by Invivoscribe of San Diego, California. We wish you all a good evening and a good rest of the conference. Thank you. This session is closed. Thank you.
Thank you.
Bye-bye.
Bye-bye.